So this is going to be fun for me because my background is entirely computational, but this is actually mostly wet lab work. So I will try and acknowledge the relevant people at the relevant points. So one of the things that my lab is interested in is this general question of how do genetic variants at the DNA level end up influencing disease risk. And we know this must happen through cellular and molecular mediators. One of the mediators we're particularly interested in is RNA splicing, hence my question. And we know that RNA splicing can result in different splice isoforms, and we and many others have helped show that that can have a really big effect on disease risk. But a lot of the time we don't know exactly why that's happening. Some of the work I did in my postdoc was to develop this method leafcutter, which is a very flexible and highly scalable way of quantifying splicing variation, but from short read data. So this is really trying to work within the limitations of short read data because that was mostly what was available at the time. The way leafcutter works is to focus on these spliced or split reads, so reads that span across a splice junction. And it builds this intron graph. So this graph, the nodes correspond to splice sites, and the edges correspond to splice junctions detected in the RNA sequencing data itself. And then we form these connected components. So a connected component in a graph is just a set of nodes where you can get from any node in that component to any other by following edges in the graph. And then we treat those components as our splicing events, and we quantify splicing as the number of reads aligning to each junction relative to the rest of the component. This gives very efficient algorithms and gives you a very flexible representation of splicing, but it's obviously limited because we'd really like to know what's happening at the isoform level. One of the things that we were able to do with leafcutter was look at differential splicing events across different human tissues. So the plot I'm showing here, the different rows correspond to different introns or junctions, and the columns correspond to different RNA-seq samples, and the colors here are different tissues. And this is just a subset of the introns that we can find where we see differences in splicing patterns across these tissues. There's many more, but are these things actually meaningful? Just because there's a difference in their splicing across tissues doesn't necessarily mean that there's a function to those differences. One of the pieces of evidence we have that's somewhat indicative that there is sometimes function is we can look at conservation. So this is looking in various different mammalian species at the orthologous introns. And for maybe on the order of 10% of those differential splicing differences that we see in human tissues, we can see conservation across mammals, implying that there is function. But it's really just an implication, right? We don't know for sure. We don't have experimental evidence. So that's a bit unsatisfying, and we would really like strategies to try and figure out which of these differences in splicing are actually functional. And we got very excited when we heard about the development of CRISPR-Cas13 systems. So the CRISPR-Cas systems you will mostly have heard about are based around CRISPR-Cas9, which is DNA targeting. Cas13, and there are some other related Cases, targets RNA. So instead of going and cutting DNA, it's going to cut RNA. And this opens up a whole host of different things that you can think about doing. We wanted to try to do isoform-specific knockdown. And so our strategy here is to target the CRISPR-Cas system to splice junctions that are specific to an isoform. Unfortunately, you can't always find one of those. Sometimes in the example I'm showing here, you can find one of those. So in this cartoon example, this 201 isoform has a unique junction that if you target the Cas protein here, then you will only knock down that isoform, or at least in theory you will. But then there's other settings where you only have a partially unique junction. So for example, between this pink and yellow exon, the junction in the middle here, you'd knock down both this 202 and 203 isoforms. So not perfect, but at least we can start to get at these sorts of questions. So one of the challenges of doing this is we don't really know what the repertoire of the isoforms that we could be knocking down in a cell type is. And that's because with short-read data, we really don't know what the full isoform structures are. I think you can guess where that's going. And then also, even once you have the isoform information, it's still actually challenging to figure out like where should you actually target the CRISPR guides. So we are big proponents now of using long-read RNA sequencing to plan these experiments. I'm showing an example for a gene we're particularly interested in, RBFOX2, which is itself a really important splicing factor in the central nervous system, and connects long-read sequencing results. So this is just showing the isoforms that have a TPM over 1. There's 12 of them. And this is pretty typical for human genes that they're this complex. You can also see some of these have the word novel in the isoform name. So half of these end up being isoforms that we detect in the Connect's long-read sequencing, but we don't find in the standard annotations like GenCode. So really important to have that long-read sequencing data. You can also, of course, you can look at short-read, and you can, in the short-read data, you can see the corresponding, the junctions that belong to these different isoforms. And I'll talk about these ones later, because these are ones that we actually targeted. But you don't get the resolution of what's the complete isoform corresponding to those junctions. So to figure out how to do these knockdown experiments, we first wanted to understand how well does this strategy actually work of targeting CRISPR-Cas13 to splice junctions. So to do this, we designed what's called a CRISPR essentiality screen. So I'll walk you through this in case you haven't seen this idea before. But we designed CRISPR guides to the junctions of a bit over 800 essential genes in A375 cells. So these are genes that in principle the cell just can't live without. And we end up, you can put the CRISPR guides at slightly different positions along the junction. And so we end up with a bit over 50,000 guide RNAs. The way these essentiality screens work is you're aiming to have just one CRISPR guide integrated into each cell. And the idea then is as you passage, so basically grow these cells in culture, if you have a CRISPR guide which is effectively knocking down one of these essential genes, that will either kill or prevent the growth of that cell. And so guide RNAs that are actually working successfully, that knocking down an essential gene, will slowly disappear from your population of cells. And that's what's being shown in this cartoon here. Ideally, we'd have an even representation of all our 50,000 guides initially, but then the guides that are actually working to knock down their target gene are going to drop out as time passes, and this is a sort of typical amount of time that you would run these experiments for. The way people typically quantify how well the guides are working is to calculate this LFC, log fold change. So this is the log difference in the counts between the initial pool of guide RNAs and what you have at, say, day 21. And you get that, you can just do short-read sequencing there because these guides are only 23 base pairs. So one place where it is okay to do short-read sequencing still, I think. This is showing the empirical CDF of guides in different categories. So targeting essential genes, targeting non-essential genes, non-essential controls, and non-targeting controls. And you can see that just for the guides targeting essential genes, you see this tail where you have these large negative log fold changes. And one of the sort of surprises to us originally was how if you just pick these guide RNAs at random, you actually don't get so many of them working. So something like 1/5 of the guide RNAs, if you don't design them in a particular way, actually end up working. So that motivates trying to figure out why some guides work and some guides don't. We actually have previous work in collaboration with Neville Sanjana's lab trying to look at whether we can predict which guides will work well based on their sequence. As I said, this is only 23 base pairs, so you can actually use a pretty simple neural network model to predict guide efficacy. This work was published last year, if you're interested. And so we were interested in training this model on our junction targeting data to try to understand whether there was any difference in the sequence determinants of guide efficacy when you're targeting splice junctions versus just targeting exons. To do that, we first wanted the best quality training data we could get. And these CRISPR screens, they're an awesome tool. They're also kind of noisy. And so to remove as much of the noise as we could from our log fold changes, we developed a Bayesian model that uses linear mixed modeling to try and estimate the slope of dropout, so how rapidly these guide RNAs are dropping out of the pool. The main contribution of this model is that it can use multiple time points and multiple replicates, which other methods can't. It's implemented in this Pyro probabilistic programming language, which is very nice, and you can write down your Bayesian statistical model, and then you just give it data, and it will do Bayesian inference for you. It builds on top of PyTorch, one of the main deep learning libraries, so you get all that good computational efficiency of running on the GPU and that sort of thing. So when we train our Tiger deep learning model to predict guide efficacy, we do get, so this is showing as you vary on the x-axis your definition for what you call essential. And this is another subtlety of this work, is that when we first went into this, we thought there were essential genes and non-essential genes. There is definitely a spectrum of how essential genes are. So as you get more stringent on what you call essential in the test set, you see better performance. So the blue here is the original Tiger model trained on exon targeting guides, whereas the green and orange are using our junction targeting data, and then this is testing on junction targeting. The Tiger bass, which is actually a fish, which is cool, it's actually a name that still makes sense. That does slightly better, so this is using our linear mixed model to get training data, but you still honestly, you do fine using the raw log fold changes as well. And so you can see there is a small improvement in the area under the precision recall curve when you use a version of the model that's trained on junction targeting data rather than just exon targeting, but it's relatively modest. And we actually, I'm not going to go into it here, it's in the preprint if you're interested. Overall, we find the sequence determinants when you're targeting junctions versus exons are broadly the same. We thought there might be effects of things like the exon junction complex, for example, but we don't see any indication of that. We compared our Tiger model to some other models that try to predict Cas13 guide efficacy. So this is showing on the x-axis predicted efficacy, and the y-axis is different knockdown outcomes, actually here just quantified by qPCR for some different genes that we investigated individually. We get better correlation than these competing methods, so we seem to do a little better at predicting which guides are going to work in a quantitative way. So going back to my RBFox2 example, I wanted to show you what the actual knockdowns look like. So I showed you that there's this diversity of isoforms already. We know there's some interesting variation at the protein level between these different isoforms. So these long isoforms that you see here, they have a nuclear localization signal. Kind of makes sense that RBFox2 would have a nuclear localization signal because it's a splicing factor. And if it's going to regulate splicing, it needs to do that in the nucleus. These, however, these short isoforms do not have an NLS, which is curious. We don't really know what those isoforms are there for, but they are there. And the interesting junctions that we can target corresponding to these two settings, this 149 junction, when we target that, it should knock down all of these long isoforms that have the nuclear localization signal. When we target this 147 junction, we're only going to end up targeting these shorter isoforms that don't have the NLS. So remember that. I've done a lot of staring at these numbers and trying to remember them. So we were actually really happy to see the knockdown results, and we were really only able to see this because of having the Kinnex data. So to set things up here, this is just showing the proportion of the different isoforms in the control sample before knockdown. And then these are showing what you get when you target these different junctions. So when you target the, let me go back briefly, when you target the 147, so this is targeting the short isoforms only, you only see knockdown of those short isoforms, and you do not see any knockdown of the long isoforms. Maybe I should say also that the x-axis here is the TPM for each isoform in the control, and the y-axis in the knockdown. So every point on here is an isoform. Conversely, when we target the long isoform, we only see knockdown of the long isoform. We don't see knockdown of any of the short isoforms. So that's great. And this recapitulates for other splicing variation that we have in RBFox2. So we only ever see knockdown of the isoforms that we think we should, and we don't see knockdown of other isoforms, sort of off-target effects. And we really wouldn't be able to say anything like that with any confidence if we only had short read data. What we can also do, since we have the transcriptome-wide RNA sequencing data, is look at downstream effects of RBFox2 knockdown. Since it's a splicing factor, we might expect to see changes in splicing, and we do see broad changes in splicing patterns across our knockdowns. This is just showing RMAT's splicing events and their PSI. And you can see that the control clusters with the 147 junction knockdown. So that, remember, is the short isoforms that don't have a nuclear localization signal. So that's saying when you knock down the isoform of RBFox2 that is likely only in the cytoplasm, there's no effect on splicing, which makes sense because that protein's not going to the nucleus where it could affect splicing. But when we knock down the long isoform, then that clusters with doing total knockdown of RBFox2, and that makes sense because now we're knocking down the version of RBFox2 that has the nuclear localization signal, and you would expect to actually influence splicing. This is just showing a couple of examples. Oh, I got the arrows in the wrong place, sorry. This is column 4, and then, oh no, arrows are in the right place. And one other gene. We picked these ones because we also have RNA immunoprecipitation and sequencing data for RBFox2, so we know that there are binding events of RBFox2 near these two examples, so it makes sense that we're seeing changes there. We have a preprint up on this work if you're interested in reading more about it. And I wanted to talk a little bit about where we're going in the future with this. So the screen that we did so far was really to sort of set up the experimental system and understand really how to do these experiments. And I think we learned a lot in that process. But what we'd really like to do is be able to do genome-wide screens of function in an isoform-specific way. And as a sort of first step towards that, my postdoc Megan's been working with a really wonderful undergrad at Columbia, Stella, who's been processing all of the ENCODE PacBio long-read RNA-seq with a slightly customized pipeline for our use case. So they have data for 96 human samples across 26 different tissues. And Stella's gone through a whole process of calling isoforms very carefully across all of those samples, and then try to look at what effects we would expect at the protein level in terms of various different features of the protein, so localization, whether they have RNA or DNA binding domains, how much change you see in the N or C terminus. And we're also looking at predictions of disordered regions, which tend to mediate protein-protein interactions. So yeah, next steps will be trying to set up to do isoform-specific functional screens using our CRISPR-Cas13 system. If you have phenotypes that you think would be particularly interesting to look at, of course, kind of the generic one would be single-cell RNA sequencing, because then you can look at lots of different effects. We're also interested in maybe looking at phenotypes like neuronal differentiation potential. Right, I will wrap up there and just thank the people who did the work. So my postdoc, Megan Schertzer, Stella, Andrew, Anjali, and Karen were all also involved. And yeah, thanks to PacBio for the sequencing data as well, which was very helpful. Thank you.